So I had a quick question about your mouse model with the EOM. So these border cells, are these originating from the smooth muscle, or you said these were satellite cells?
So this is a very good question. I didn't get to it because, you know, Hugo was looking at me. I knew I better be, I don't even know how much time he allocated for me, but I saw time. So that was my biggest achievement for the day. So what happened is some years ago, because of my interest in parasites, again, somebody told me, you know, there is the smooth muscle myosin SM11 Cree your parasites, you know. I thought, well, we will be able to sort them based on this. And that is a very interesting construct because it also has its own GFP. So you can rely on its own GFP versus amplifying a GFP from ROSA 26 and TMG or all sorts of things. And I don't remember how was it Andrew that looked at the EOM? How did we suddenly saw that it's not just that there is, it's expressed in not where expected? So immediately the first thing would be, wow, finally smooth muscle cells are making skeletal muscle. We got the proof, wonderful. But as we sorted, and it was Andrew's work, he went all the way to time 0. We never went earlier than that, but he had to collect a lot of babies. It wasn't trivial. Luckily, the sorting was still free at that time. When they started charging for sorting, we stopped doing experiments because that became outrageous how much it cost. So we did cytospe, all sorts of things, and those cells are PAX7 positive. Okay, so what we, in the first version of the paper, there was a lot of elaboration how mesenchymal cells became PAX7 and so on and so forth. I just think that what we needed and was not available then and available now at the department, but I don't have any more the grants to do it, there is a microscope that you can do pretty thick sections. And instead of working with GFP, we could work with the LAG-Z, the nuclear LAG-Z. David Goldheimer has a fantastic reporter, which we used, it's nuclear, and start going earlier in development and really try to track the whole three-dimensional structure. When does it happening? When those cells are first expressed? I cannot comment about anything earlier than day 0. But at day 0, the satellite cells, and we have a lot of pictures, are PAX7 positive. So I think it would be a far-fetched to think that these are coming from smooth muscle. I think that either there was a minute of expression of the smooth muscle Cree, very transient, or more so, maybe it's a transient, the Cree. Maybe it's not 100% just smooth muscle, and or maybe we stumbled on another lineage that is so beautifully in the EOM because there is a total match with the development of the orbital layer in the EOM. So in the EOM, most of it is a global layer, but then it has an orbital layer at the external periphery with it's more embryonic, it has a whole different gene expression. And the thought is that that layer is very important for our vision because the extraocular muscles need a very minute control of different activities. And way back, I knew more about that layer. I've been away from the EOM for some time, but it's so beautiful because the other papers talk about changes in isoforms, in myosin. And here we are telling them, wow, this is not just happening like that. There is a satellite cell population coming in and generating the orbital layer. So I don't know if this addresses your question, but I don't think it's smooth muscle. But another point that I didn't make is that the smooth muscle, extraocular muscle, has a lot of a smooth muscle system called pulley. And again, I give all the credit to Andrew, who did a fantastic work and even spoke with pulley person that does MRI of the eye pulleys and so on. That entire thing is very positive for the myosin heavy chain 11-3, but that is a bona fide smooth muscle. So there was a time I thought in the lab that maybe the cells from the pulley are migrating to the external side of the orbital layer and becoming satellite cells. I basically believe in simplicity in biology, and it will be better to find a different explanation than smooth muscle becoming skeletal muscle, but it's not out. We haven't ruled it out. Any other questions to Zipora? Pascal? Any comments? Pascal was very involved in the EOM work. Maybe he has a comment to say.
No, I just had a comment regarding the spindle. Because yeah, one day I still want to, I'm still interested in the spindle, but I don't, are you aware of any paper showing any regeneration of the spindle?
You showed. We once injured with cardiotoxin.
Yeah, I remember we had
yeah,
kind of glimpse that it could happen, but not really proof.
I think that there are papers from Sweden. What's his name? He's the one that had in his team the woman that didn't like us.
Okay.
Tornell, he start with a Torn. He has some regeneration. I think that the spindle field is a gold mine waiting to be explored now that there are so many reporter mice. And many physiologists study the spindles. There are huge, the body of literature about spindles is huge. A lot of it is done in the cat, for example, because they have, the cat has much larger spindles. And I think it's a gold mine. There was a guy who did some developmental biology way back. He even had a mutant mouse that I don't know if created more spindles. He had an NIH grant to study it. I haven't followed more, but one of you guys with all your talents got to get to the spindles. You know, I'm talking now like an elderly lady. I remember when people used to go into labs and visiting and say, you know, why don't you study this? Why don't you study that? I did all the pilots, go ahead. I don't want to sound like an elderly lady. I'm just thinking, I have a hunch that whoever gets into spindles will be close to the truth more than ever before.
Christoph, your mouse that died after 5 days when you kind of ablated satellite cells, PAC7, you put it down to spindles or neuronal issues. Did you ever resolve which?
Yeah, no, we haven't resolved that. I mean, the time frame is really a challenge with our mouse model, right? Because they die so rapidly. But one thing that was interesting, and I have the labeling behind me, but it doesn't show it here. The one structure that we always labeled with the LAC-Z PAC7 382 was a spindle, even without injury. So there clearly is interesting satellite cell lineage activity, differentiation activity in the spindle happening, but we have not followed up on that.
I'll comment on that, because that one is weird, right? Because we can see the spindle labeling very quickly after labeling the PAC7 within the spindle. If that were true, that means spindle is constantly remodeling. So that one is the one I'm not sure. I don't know whether Gabrielle's Cree does something different. Because I was told by Patrick Seal there's some difference of Gabrielle's Cree R2 and ours in labeling different cell types.
Before Gabriel answer, I would like just to say that the spindles fibers are so tiny. Their diameter is so tiny, even smaller than the EOM. So if you find quickly a label there, I don't know that it suggests that there must be quick turnover. and that's back to the work that Gabriel published on the different muscles. When the fibers are much smaller, we need to be careful because the label, if it's not nuclear label, gets there very, very fast. But on the other hand, I learned from Sharagim in a discussion with him, and then I reviewed the literature, that if you express PAX3 as a satellite cell, you are more under stress. And there is a work from Frederick Kreilik's lab and a few other labs. So we need, maybe when we think about spindles, we need to think about also the PAX3 that is so easy to detect in some of the satellite cells there. Are they proliferating? Who knows? But without, I don't think we can survive without spindles. So it could be that the mouse of Christoph and Fan died so quickly because they targeted the spindles very quickly. Even the envelope of the spindle expressed PAX7, that was shown by a group from Australia a long time ago.
Charles' mouse, the MIF5N-LAC-Z mouse, when you make single fibers and you stain it, all the extrafusal fibers, satellite cells get stained, but the spindles light up beautifully. It's a really nice way to get out muscle spindles and look at the structure of them, just to isolate them. It's quite hard just to identify them when you do single fibers.
You
but
published that.
No, exactly. But to really, and the structure is just amazing when you see them as single fibers.
So just to remind people, there is the capsule, and there might be 4 or 5 fibers in the capsule, but then there are the extensions out of the capsule. And if you do a cross section, maybe you hit the extension and only 2 of them are there. So it looks like glasses, like a pair of binoculars. And I pointed out to some of this in my, Miranda Grounds and me were planning to write a book, but it just, there is always something else to do. And there was recently a review about spindles published in Skeletal Muscles. So we kind of felt like, are we going to duplicate that? Or, but there is a tremendous amount of literature out there on the spindle structure, on the innervation. To me, another big question is, how does the body decide your fiber is going to be a spindle fiber, or your satellite cell is going to generate a spindle myofiber and not a regular myofiber? Maybe Sharagim, with his fancy collection of mice, can comment. What can we do?
So actually, I was going to comment that Carmen Birkmeyer, who's done, multiple labs have done the single nuclear RNA-seq. So Carmen has pulled out the spindle signature, and it looks like it's not so much muscular, but it has some neuronal features in there as well. So it's going to be quite interesting, and there's some unique genes that she's picking up, and she's following that up. So I think that's going to be fun to look at when also spindles from different regions. I mean, she took it from the hind limb, I think, or maybe even the TA, probably the hind limb entirely. So regional specification of the spindles might be following regional specification of the peripheral nervous system. We'd have to see what that gives. But I had a question for you, Zipora, and you, Gabriel. And it's an annoying question that Zipora, you get annoyed by this. Proliferation of extraocular muscle satellite cells or diaphragm satellite cells. So Gabriel has done this, and Brad also have done this nice lineage stuff showing that the diaphragm, the EOM, have a higher turnover. But for the love of God, we haven't been able to pick up BRG positive cells in the adults. And I know that, and you don't either, right?
Shall I tell you, I love you. You don't annoy me. Pascal can talk about it. We bought a cytospin machine because we did so many cytospin. We got tired of going to other people, and we published it. And we were pretty afraid of Linda McLuhan, who showed the model of building fibers, continuous. We don't have any evidence with BRDU or with Ki-67 that the satellite cells that we harvested from the EOM, and Pascal can talk, was it Andrew or Pascal?
No, he did it also, yeah.
We couldn't have any evidence, and we published it with great fear that somebody will tell us something is wrong. Luckily, we published it. So we are on the same page.
So we're on the same page, but I think Linda McLuhan's data is, she's picking up other cells, because we went back and looked at her CD34 positive cells, and it's not just PAX7 GFP. They're picking up all kinds of other cell types in the CD34. But then, so if we cannot capture cells dividing, how do you explain the contribution over time, the high contribution over time? Is it just they're sneaky and they're going under the radar, and when we BRDU labels, just decide to shut off, and then, or they go in bursts?
That's a question, I think, to Gabriel, not to me, because now we are talking about her results with the green
contribution
fibers.
over time, right?
Yeah, no, I agree. Like, do they, I mean, I just wonder, do they just literally just drop in, right? They don't divide, but then, right? Then you should be depleting over time,
right,
the resident satellite cells.
So there's an odd result that got buried in our paper with Philippos in 2012. When we inactivate RPPJ and Notch, satellite cells wake up, and Tom Rando's lab saw that too. Okay, fine. But when we add, inactivate Notch and add BRDU, they fuse to the fiber and they boycott S phase. So they just skip. They go from G1 to myogenin. 75% of them do this. And it's the first demonstration in vivo of a boycotting of S phase coming out of G1. And people have seen that extensively with SV40 antigen, et cetera, but under homeostatic conditions, it wasn't seen. But this was when you remove Notch activity. So that means you can, as a satellite cell, because that's the paradox, you need to get rid of Notch to activate, but you need Notch, according to the Fly model, to negotiate G1S. So it goes without Notch, it wants to activate, but there's no Notch, it can't do G1S, so it boycotts S phase and it fuses to the fiber. So we do the lineage tracing and we see it fusing to the fiber in the absence of BRDU. Now, what's interesting in the control mouse, in the mutant Notch mouse, 75% of them were doing this, boycotting S phase, but in the control mouse, about 8% were doing it. So what you're saying, Gabriel, is perhaps in EOM, there are more cells that boycott S phase. They wake up, they don't divide, and they fuse.
Right.
But then if that's the case, you need to see depletion over time.
Yeah.
Right.
Much faster, but you don't.
Right.
So there's a red herring here somewhere.
Yeah. What about Charlotte's Peterson model of injecting?
Well, if that's true,
injecting without fusing,
then you have to have many more vesicles produced by the EOM than by the TA. So that has to be checked then, right? Are there many more exosomes or vesicles that are picking up the tomato or the lineage marker, fusing
need to be much
in the
more.
EOM? That's possible.
The EOM are so narrow, so tiny. So if the satellite cells release the same amount in the TA and in the EOM, you will catch the color much, much, much quicker in the EOM because the diameter is so tiny. So think about 10 molecules in a room versus 10 molecules in a jar. Yeah. So I look at the EOM as the jar and the TA as the room. The concentration of the 10 molecule is much higher in the jar, even though absolutely it's the same 10. So,
yeah, the devil's in the detail. We still haven't closed the chapter on some of these things.
Go on.
One thing I found quite fascinating from today was that idea that you can have a set number of satellite cells and it's lower during development. So I can understand that, as they proliferate, your control. But then when you regenerate the muscle, I think it was the FGF6 knockout, when you regenerate the muscle, you then get less satellite cells back again. That is really strange because, I mean, you're only one division away from having the correct number if you've got, I think it was 40% or so.
Yeah. So
I understand as it grows up that, it's quite coordinated and that you can have less satellite cells just because you're not stimulating with the T. I'm sorry, I'm confused which one it was. But it's just when you regenerate, it's so chaotic. that you then maintain that reduced level of satellite cells again? I thought that is just quite hard to explain.
I have a different explanation for that because FGF is expressed by the fiber.
Yeah.
So the regenerative fibers still have less FGF6, so they couldn't go that extra cycle, just like during development of the 14 days, right? That's 40% difference. So I think it's just the same thing. You are generating new ones, but the new one is still deficient of FGF6, so you can never catch up, and it's always one cycle off. That will be my interpretation.
But you're going through so many cycles to generate that.
I really think the end, I think that part I agree with Sheragim, because I really like Sheragim's paper on when do they become quiescent and reestablish homeostasis. I think it's rather late. We all seen like 21 days, still more than without. It actually goes down further with the time frame. So I don't know exactly. I think Sheragim said what, 3 months? They're still not quiescent.
Well, there's still some signs of activity. Yeah. We haven't gone that far.
So I think
one of the things, sorry, if you look at the different injury models, if you start with 7,000 or 10,000 TA cells, you double that after 3 weeks, and then it starts to reduce. Exactly. So we don't have the kinetics of that. But if you're double that, that's only, as Pete's saying, 1 division. So then if you go down with the FGF, then is this is going to be heretical. Is there going to be a mitotic clock there? You know, we've gone through that in the field, right?
You really don't know where do they. I mean, I think everyone wonder where do they go, right?
Yeah. So back to the FGF6, we worked on it in 2000, and we showed that the FGF6 was in the fiber. So at that time, we didn't look at satellite cells. We did fiber versus the rest. The rest was mostly, you know, we isolated single fiber. There was a very good protein level of FGF6. We added all the FGFs to FGF6 null fibers. We didn't publish it because I was very disappointed with the mouse. You know, now Christoph is finding finally some differences, but when the mouse was originally published and you looked carefully at the paper, it was 1 RT-PCR defect in the region, you know, if you look at those profile. So he needed here 24 cycles, but here 25 or 26 to show the gene. I didn't feel that was talking to me. I think that just like what I asked Beth about IGF-2, my mindset about the role of FGF6 is different because it's so myofiber specific. Maybe we are still, so when you found a decline in numbers earlier on, what does it mean? Just like what Peter asked, there are so many cells, so a little bit less, little bit more. The mouse looked very happy. We did a lot of analysis of myosin, slow, fast, troponin. We didn't find any. They were running. One of them even jumped from the bench down and we couldn't even find him anymore. And then a month later, a friend told me she found the mouse. They were C57 Black Sea wild. I didn't think there was anything different. I was even disappointed I brought them here because it cost me an arm and a leg to import them from there. So I'm very happy that you are finding a difference. You know, there was another FGF6 null mouse made by the French. And when I met the French, they laughed at me and said, oh, you got the FGF6 null mouse? Good luck, try to find. Eventually they published something about the soleus. That there they found a difference, and maybe because it's so slow, such a slow muscle. Maybe you should consider getting that mouse if it still exists. Who knows? I don't want to overload on your work.
Yeah, but you can't forget this. There's a lot of technical differences in the studies, right? So Thomas Brown's original one, he did the cryo-freeze injury, right? And he has also histology. He didn't just do the expression analysis. He also has histology, and it's actually a severely compromised regeneration defect in his hands. But then he did the single fibers, and back then there was no PAC7, of course, so he did CD34 for some quantification, and he didn't pick up a big difference with that quantification method, right? But yet the regeneration defect by histology was massive in his hands, and so I believe it's likely a lot of it is technical difference. So we do the cardiotoxin, which is more similar to what the French group had done, and they didn't report any, as you just said, any regeneration defect in the TA muscle. But I think we also only have a very subtle defect, right? So at 3 weeks out after injury, they are now that we are able to more carefully quantify myofiber size, we picked that up, but they didn't back then. So I think a lot of that is some of that is technical in nature too, and it is a subtle. Somewhat subtle decrease in satellite cell number, 40% reduction, but it's incredibly robust. We see it in every muscle group we analyzed.
Myonuclei.
I agree with Pete. It's actually surprised. We were surprised too that it wasn't reset during regeneration. That
what about the myonuclei number on single fibers? Because if you're lacking a division, is that a division for self-renewal? Is it also one division for regeneration? And if that, you'd see a major difference in number of myonuclei per fiber, surely. Did you look?
So we haven't done it in whole mount fibers. We've done it in sections and then normalized for fibers in the TA. And by that method, we don't pick up a difference.
Yeah, but again, I think it's a similar, taking a section, I think isolate single fibers and DAPI and just count DAPI pack 7. And if it's an EDL.
Easier said than done. It's a lot.
People are good at that.
It's quite consistent. There's roughly 270 myonuclei in an EDL fiber. It's not plus or minus a lot, 20, I can't remember exactly, but that gives you the absolute unit. And if you're talking about just divisions and there's being less satellite cells, you want a really accurate number of how many myonuclei are there, just to see if the division is purely a renewal, self-renewal division, or also differentiation type renewal.
Oh, good point.
It's a lot of work. Christoph, there were early papers where they did in situs for FGF6 and FGFR4. And during it was a mouse paper. During development, there is a complete co-localization of FGF6 and FGFR4. And they are both very specific to the muscle and not to the other cell types. So the thought was that maybe FGF6 is the specific ligand for FGFR4. I don't know if you are going that direction or not. We tried it, and it could be that when we ablated FGFR1 and only FGFR4 was left, we gave FGF2, but maybe if you give FGF6, you will finally figure out the interaction between FGF6 and FGFR4, and maybe that's a very specific ligand for FGFR4. That was the thought way back, but there are no physiological experiments to prove it yet. So I'm just saying that look at the early papers where those two are the only two that are really co-localized and muscle specific are FGF6 and FGFR4. And
I think Hornets also tried the FGF6 and FGF2 and showed that both of them will bind to either R1 or R4.
But that's test tube. That's ligands. That's test tube studies. Everything was done in vitro. The amounts, who knows? But I think you might end up with the same headache about FGF6, the headache that I got with FGFR4. I hope you don't, but I definitely spend a lot of years. And I'm just hoping that you will solve it, because that will be a great endpoint to my misery. Yeah.
No, clearly I think we've been profiting from much better tools now, especially as you mentioned, PAC7 was a game changer in our field, and we have the ability now to very carefully quantify satellite cell numbers and really pick up these differences. And then again, the FGF6 mutant with a 40% reduction doesn't add up to a huge impact on our regeneration assays, although we also have significant decrease at 3 weeks out. but nonetheless, it's almost one cycle that the satellite cells, one less cycle the satellite cells undergo during development to generate that pool, that reduced size pool, right? And clearly, we've been profiting from these strong tools now to do some careful analysis for these counts.
So what does it mean once one division less? Is it Howard also believed in the quantal mitosis, the one that after that there is differentiation, and there were fights in the field. They were ready to kill each other. I was very young, and they say, lucky you came from Yaffe and not from Holzer and not from Königsberg, but at meetings they were killing each other about this model. So if there is one division less, is that does it have a meaning, that last division? And if they don't have this last division, they are missing something. Maybe they cannot differentiate as well. Just not dividing one more time, it's too trivial. I'm not saying it against you, but there's got to be something associated with that one more division that is missing. What is the missing player now? Because otherwise, who cares? One division more, one division less. Big deal.
So Zipora, this is a question that's keeping me up at night, and I have to run, unfortunately. But all the tools that we have in the market, except N14, C13, and maybe EDU at a very high resolution level, all of them allow you to follow a cell only for 5 divisions. After that, you lose your markers, BRDU, H2B, GFP, etc. So if everything happens within that 5 divisions, it's great. But once it goes beyond the 5 divisions, you can't track the cells anymore. And then you have to come up with models, which is a little bit frustrating. So until somebody who can get us a trackable quantitative mitosis for multiple divisions, I think that's one of the going to be the big additions in the field, not just for us, but for anybody.
Thank you.
So on that thought,
that's one of the biggest challenges of the field, I think.
Yeah.
The labeling of more than few rounds of division. Goodbye. It was a pleasure to have you here.
Pleasure all mine. Great to see you guys.
Bye. Bye, everyone.
When is the next in-person muscle meeting? Is it meant to be the Costa Rica meeting? Is that still on at present?
Is that your meeting? You were the one organizing it.
No, not this. Yeah, a lot 2 years ago, but not this year. It's Saverio Tedesco and Rita Pellegrino and Elizabeth Chen are organizing it.
Closing the day.
Yeah.
Sorry. I need.
It's meant to be in November, I think.
Yeah, and for now it's still maintained as far as I know. But...
Just in time, but since I am in one hotel, I have to wait. I asked for privileged service, but not completely. Anyhow, I am happy that to be here and congratulate once again with Zipora for his wonderful suggestions and patience, because we exchanged 2,000 emails.
I saw 2 million.
But it was a wonderful winter. Then Zipora was upset with Ugo, which is not difficult to be upset with Ugo, and said, I will call you 2 days before the conference, this before Esther. You remember, Zipora? Or you forget?
No.
I am happy that you forget.
No. So you had,
nice to see.
Everybody is leaving, only few people are left. Maybe before, Manuela, before you leave, did anybody have questions for Manuela? Because she had a lot of new data to the field, very interesting, but there was time only for one question, I think. Anybody has questions to Manuela?
I'm going to have to watch her video on YouTube. I was having blood taken.
Manuela, I don't know how she contacted me. if it was through friends of Zipora or independently. What do you think about?
She contacted you she saw I gave you her email. You send her the advertisement.
The program.
And she contacted you independently because of her Italian loyalty. And then you told me who contacted you back. And I saw that Manuela already did it. And this is because of her Italian roots.
I was hoping to be in Italy. I was hoping to be in Italy in person, but unfortunately,
she is open to return in Italy, but it is the chance is almost to zero, unfortunately.
Yeah. How is it in your place in Europe? Here in the United States, people started flying, but within the States mostly. Canada is still closed at the border.
No, here, if you have a job motivation, you may fly in Europe. You agree, Peter? Not, but England, Great Britain is no more EU.
No, we're busy developing new variants for export.
They decide that it is better to stay alone. I don't know. I'm not sure. I have a son living in London with 2 children. It is 1.5 year that we can meet, and maybe this August there is a chance, but I'm not sure.
Yeah, I'm finally going to see my sons in July. I haven't seen them since December 2019. You know, I went to Israel in February 2020, and the virus was just creeping in, and I asked people, what shall I do?
No, but I am confident that vaccination will do.
There is no question about it.
It's part of the solution.
In America, it helped tremendously. People are living again, and Israel is the model. I think they are removing all restrictions next week.
Yes, but
in
Israel, they are 11 million.
Yeah,
in India, they are 1,200,000,000.
Okay,
and the situation is horrible.
But Israel is a model, a laboratory to show that if you can immunize a large number of the population, you conquer the disease.
You imagine what means to make shots twice to 1,300,000. It is 6 times the states. It is states, Europe, and maybe half of Africa alone. Anyway. 1 million are Chinese people, another are in the South Pacific area, 300 million are in Africa. How much in South America? 200 million or more? We have to wait that all are vaccinated.
I'm glad we're recording this.
If we are lucky, that mutation will be mild, minor problems.
We can
Anyhow,
I
have my red wine.
Okay.
And this is to make chin chin. Auguri, I don't know what you say.
You had a question to me, to the muscle people. We are still 5 muscle people, so it's the last chance. Maybe you can ask.
The last time. I was not shocked, but Terence Partridge remember us that, and someone added that the satellite cell may stay at 4-6 degrees in a death body for up to 30 days. You remember? Someone remember?
Yeah.
It means that they are like non-oxygen bacteria.
Aerobic, yeah.
They have a metabolism, non-oxidative metabolism in that situation. Who is interested to check PubMed if there is results on this. Because if there are no results, this, in my opinion, this is a biological problem, incredible. Why these cells, the only rationale is that if you have artery constriction, artery occlusion of a big muscle, you will have 1 kilo, 2 kilo, 5 kilos of meat without oxygen for at least 1 month to have growing capillaries everywhere. They are programmed for these events, but this is not a solution because they are not life-saving events. Satellite cells have to be, the same is for the very, very, very rare mutation of the genome important for satellite cells that do not produce frequent myopathologies. There is maybe one published, Gillian Battle Brown sent to me a message, yes, there is something essential for the satellite cell survival. If there are mutations in those, I don't remind what, there is a very rare disease related to the dysfunction of the satellite cells. This means or the mutations are lethal, but ought to be lethal much previous than the job satellite cells make in muscle. You begin to understand the problem, to find a solution, a rational explanation why these cells are so tremendously resistant, except heavy irradiation. They are like horses during the winter. They stay there up to the sun is warm enough.
But how, if they are so quiescent that they don't notice somebody's dead for days, yet you can activate them in how many hours? I mean, it goes from being ridiculously...
But how could they survive 30 days in a cadaver?
Yeah, no, exactly. No one's told them the person's dead.
They have to be glycolytic, able to extract energy from glycolytic molecules around the body, everywhere, including when they are in the dead muscle.
And also, we are not talking about the whole population. It's certainly just a subpopulation of basal stem cell, not the whole population. So yeah, I'm seeing the same as Pete just said. I'm just seeing it as it's just deep quiescent cells that are here to maintain the long term. And yeah, they are.
How many mitochondria there are in satellite cells?
How many what?
Mitochondria. There are a lot of mitochondria? Not.
We don't know. I don't think there is literature about mitochondria in quiescence.
But why no one are interested? This is a major biological mystery.
So I actually want to add something that I meant to talk about. When we did the FDB fibers way back, there was no theory about stem cell and asymmetric cell division and all this. For me, the idea was, are there quiescent cells that don't wake up at all with all the manipulation? So together with Tony, we gave cytosine arabinosid that kills proliferating cells. The cultures were going on in that restricted medium, you know, it was serum replacement, or we gave FGF. And the idea was, after a few days of cytosine arabinosid, we will extract the whole thing and put it in very rich medium and see what survives. And whether it was, whether it was with FGF or without FGF, so it was those plates that we had 30 fibers per plate, FDBs. In each case, there was one colony in very rich medium. So in that rich medium that we described for our primary cultures, everything grow. In each case, there was one cell. I never published it, but those were deeply quiescent cells that being exposed to the basal medium. So it's like the dead body that you are describing, I think, because that medium was very restrictive. If you remember, they went very quickly from proliferation to differentiation. So maybe the satellite cells, a very small fraction, in our case, it was one cell, can survive very harsh conditions. Does it mean that it's a different population? You know, the people that do single cell analysis, they can address a lot of questions.
But are you sure that there are not mitochondria in proliferating myoblasts? Are you sure?
I don't know anything about mitochondria, so I can't comment.
You have seen millions of pictures in electron microscopy, or
there's definitely lots of mitochondria in myoblasts. Proliferating myoblasts have mitochondria.
Where? In satellite cells, there are no mitochondria, even when they divide.
In the progeny, yeah, in the satellite cell progeny, there are lots of mitochondria.
In the myoblast, the proliferated myoblast.
Yes.
But not in the quiescent satellite.
We don't know. We don't know. We haven't. It's such a tiny cell. But, you know, it's so tiny that. But you look at the EOM.
There are more people studying mitochondria than people studying satellite cells.
Yeah, but
there are
millions. There is not a single
trillions of pictures of mitochondria in any tissue, except satellite cells. I can't believe.
Not a, you know, all these people that look at mitochondria in skeletal muscle don't look at satellite cells. It's such a rare finding. You know what you need is
because they are not curious. I am a curious person, and sometimes I have a good idea on this crazy thinking.
So you think maybe they survive?
I would not convince you that it is a good idea to invest money, work of people, and so on. But it is an interesting biological problem that could be discussed next year, hopefully, in Montegrotto, with a glass of wine in one of your hand, looking at the sunset on the Euganian hills.
Bischoff wrote in one of his reviews, and I can never find it again, that the entire activation of satellite cells is tension, electrical tension from the membrane of the myofiber. I wish I found it again. I can never figure out, because you said, Peter, how can it be so quiescent and yet activated just the moment you start cutting the tissue?
I can't believe it. They are there, and actively they stay there up to the moment there is a minimum irritation, minimum damage to the muscle. The most strong evidence of some myopathy is that the nuclei go from the periphery to the center of the fiber. This means that there is a mechanism that maintains normal nuclei, a satellite cell, active mechanisms along regular life of the muscle. Because if you disturb the muscle cells, the muscle fibers, the nuclei move in.
I think that Tom Rando talked about that the quiescent state is not a quiescent state.
They are not stick.
I don't know.
They are stick there. We are
now going to, unless my colleagues have something to say. Do you have another question? You said you had two questions.
No, two questions. One was survival in anoxia for 1 month. The second was this idea of I don't the second was related to we are all very tired.
Okay.
It might be time, I feel.
The first question should be addressed to maybe Terry. He is the one who isolated.
No, the second question was why there are no myopathy as frequent as Duchenne muscular dystrophy, which is dependent from a gene mutation of the set of gene or promoter or enhancer, whatever you like, relevant for the stability and the functionality of satellite cells.
I have the answer.
Quiescent or activated.
Earlier today, we sent you a review in your email about a review we've just written on satellite cells and muscular dystrophy. So we listed about 3 or 4 diseases. There's one called MEG-F10 that affects satellite cells. There's a MyoD mutation that has a satellite cellopathy, and they all seem to be congenital with scoliosis and breathing difficulties. There's a PAX7 mutation as well.
Yes.
So we sent you
are
the review.
Beginning to describe something that could be described next year, if you like.
Yep.
If you have, if each of you have a much more interesting topics, we may discuss. Of course, I have the key of the computer. I am in a position in which I can choose the topics of the conference. But I met Zipora 30 years ago or more. Zipora.
We met, I think, in 1986 in Tiberias.
1986. We were pen friends for 86, 15 for 35 years. Last year, I invited Zipora maybe 10 times or more.
Or more.
More. Unfortunately, I am not a rich man. I am dependent to sponsorship that disappeared with the pandemic, and I am very grateful for each of us to those that provided a minimal donation. It is not the amount of money, is the generosity, is the evidence that people value what we are organizing. This is the reason why I asked a minimal support. Just to say, are they really interested? Interested to a level to spend $100 to register? But $100, $200, $300, $1,000, $2,000, $2,050 is more than nothing.
You know, Pascal is happy to come to visit Padua. He lives very close by.
I did this only to very good friends, old or new. But let me say that for me, this is the real evidence, not only that there was every day 40 people, the 40 or 50 the first day, 20 of that disappeared and were present in the second day, up to 50 again. Today we were 50, more or less, but only 10, 15, 20 were present the 3 days. Tomorrow, no one of you will be interested to balneology. Maybe you are curious
Maybe
to
we
know
are tired,
what it is.
But Fabio, I'm saying, Hugo, there has been, the Zoom meetings have been all year long, and you know, I become more selective now. And
you are right, me too.
It's getting to a point, so if people don't join sometimes, it's because they need to be more selective, because otherwise you can watch Zoom 24/7.
Yes, you have to be. You have to have 10 computers in your room and jump from one to the other, because there are minimum 10 offers in the same moment.
But anyway, let's finish. This was a wonderful opportunity for the muscle community, for the myogenesis people. For me, it was a good event. I felt that I was missing it because I retired during this year, and here I have all my friends and colleagues and even former lab members. And whom do I know the oldest from the relationship is Peter. I will tell you a story and then we finish. Peter put a poster out one day. He was young, ambitious. I came to him, I was full of myself. And I said, where is my reference? You did not refer to my paper. I've never done it to anybody. And since then, Peter gave a lot of respect to that paper, more than me even. So thank you, Peter. The 94, you remember, you kind of stood by your poster, and I just realized how aggressive I was, and I'm apologizing so many years later.
I still have flashbacks.
You still have the flash. You probably have a deep down.
Because that was the Wiley meeting in Washington, was it? It was in Wiley, I remember.
I don't remember where it was. I just remember that
it was the first time I met you, and this probably had some resonance. And you were giving a talk, and they were 20-minute talks. And you stood up and gave your talk, and the timer went, and you just finished the introduction, and you said, "What? It's time to do the." I was just remember that sitting there. You spent the whole 20 minutes of the talk just giving the introduction, and then the timer went.
May I spend my last? Open my heart to new friends. I am an editor of a minimal, irrelevant journal, which is European Journal of Translational Myology, which is recognized by Scopus by 2.1, which is better than nothing. I asked Zipora, why not you collect 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 communications for this journal? 2,000 words, some rubbish you have in your on your back, because this may increase the audience, and attracting one good article per issue, it is essential to survive. I take the liberty to ask you to invest some minimal effort, some minimal data you have somewhere that are not astonishing, but it is surely much better than the mean results I am able to attract. Think about and send it to Zipora, and Zipora will edit the special section.
Already giving more work. We all have the,
you have to say yes. I will do the job. As usual, I will be your slave. But I need someone that have a good relation with young people. Only you are able to oblige 1, 2, 3, 4 of these people that are staying here now.
Let me tell you what's
make a minimal effort. You will work one day, half an hour.
The young people look at impact factor. The first thing they ask, what is the impact factor?
The brilliant young people invest in the future.
All
invest
of them
a minimal effort on a growing, because it is 30 years that I make this journal to survive, which is not easy at all, because there are 1,000 journals every week. 99.9999 disappear in 6 months. I have no other argument to spend.
My forest team colleagues, If you want to contribute something to an issue, we can even do a following up of this meeting. One of you can be a
Peter, you have to convince Zipora to spend her name and we will do the job.
I spell my name. My name goes very far, very
May
easy.
I spend, I have 1, 2, 3, 4 people that listen, that Zipora is open to be the guest editor of this small or long series of myology related.
I even have an idea what I can contribute if Christoph is okay with my double null R3, R4 spindle story, you know, body size. This is not going to interfere at all with your research, and all the data is ready, but we can talk.
But you have not to spend the best data. You have to spend something that you do.
I don't have best or not best. I go by the audience.
You have to be the guest editor. You accept to be the guest editor. It is more than enough.
I am not going to twist people's arms. I had a lot of work.
If you say no another time, I will ask Peter Zamet to do the job.
I would say
and have
one of the 4 guys on this panel are going in with me.
This is Mediterranean negotiation.
I need one of the 4 people to go with me. And either we draw a luck and see who is coming out, or I need another person. I don't want to do it myself alone. Okay.
I don't ask you to contribute. I ask you to allow us to write special issues on satellite cells 2021. Guest editor, Zipora, Yablonka, Zipora, Rewini.
Zipora, Yablonka,
It is a big job for you.
Yablonka, Rewini, double hyphen. Anyway, remember how it was with the meeting. You only wanted me to invite people. I'm not objecting to do it. I just don't want to be alone. So if you want to use my name, I need another person with me.
I wouldn't oblige all these people stay with us, Zipora. They are free to disconnect the connection. I'm sure that they have something more important to do. And
one more thing, that in our field, we saw the Gordon research conference this year postponed or even canceled. So this was fantastic. And I really appreciate both your effort, Hugo, and Zipora, because this, keeping the scientific exchange going is really instrumental to science, and so it's really much appreciated. I know how much work
I am very grateful because it is I dream all the days, but I need someone that say what you did, that the effort to organize this with the help of Zipora and maintain the time in reasonable scheduling, it is worth to be done.
Indeed.
Next year, I hope that at least half of you will be in presence. For sure, half of the speakers will be virtual, because it saves time, it saves money, it saves time, particularly the virtual meeting, but I hope that some of you are curious to return to Venice, to see the Colliugane, to see Padua, which is a wonderful
Let me tell you,
city.
If there is no virus,
March 30, April 2, because Easter next year will be 17. Therefore, the hotel is empty. Very cheap.
Passover is, we don't want overlap with Passover either.
If you have big conferences in those days, please let me know as soon as possible.
Okay.
I have to start with the date so that people think if it is worth or not to engage.
Okay. I think maybe one of the young people will write me an email that they are happy to be guest editor together. Then it will be a good solution.
I spent my best to convince someone to do this. You have better option. Of course, it is rational that you invest your time in better option.
I don't go anywhere. I'm very loyal to you and the journal. You know that. I just want to have another colleague with me. Okay?
You have 1, 2, 3, 4 people here and other 10 around. Writing a mail, you make ask 2,000 people. It will be not difficult to find this.
You know,
second guest editor.
Some
Peter,
like
are you open to help Zipora?
I'm not a young person, unfortunately.
You are not a young person. You are much younger than me and than Zipora. Therefore, you are young. We are young too. You agree, Zipora?
We are young at heart, and I don't know what age you are, but I am very young at heart. Am I? You all know me. Okay.
Are you curious? Would you have my age, to know my age? Put in your agenda 2023 to congratulate Ugo for his 80 years.
Wow.
Buonanotte.
Okay, as they say in America, let me think of this. In America, they don't give answer right away. They say, I'll think about it, okay?
I know you, I know you since 30 years. I know that it is enough to insist 1,000 times, then the 1,000.1, it will be C, yes.
It worked, here I am. I organized a nice session. It worked very well.
Va bene, grazie.
Thank you very much, everybody.